All rights reserved. We reserve all rights. Iatrogenic means death and injury caused unintentionally in a medical patient by a physician's words or therapy, even if that physician treated the patient exactly as they were taught to in medical school and further training from pharmaceutical drug reps. The entire allopathic field of modern medicine, pharmaceuticals, vaccines, infectionology, bacteriology, and virology is nothing more than a for-profit iatrogenic death cult, tantamount to a dangerous worldwide religious cult, causing endless human suffering and misery by propagating long-ago debunked myths such as bacterial infection, biological germ warfare, and contagious viruses, all based on the never-proven germ theory. The tentacles of this trillion-dollar-a-year cult even reach as deep as Hollywood, pumping our heads full of frightening blockbuster dystopian films about these disproven imaginary viruses wiping out all of humanity, unless our heroes at the World Health Organization and the Center for Disease Control can race the clock to find the source of the single monkey or coconut responsible for wiping out the entire human race. This is, of course, absolute nonsense and propaganda. It is time we all learn the truth. Germ theory is just a fictional theory, like the existence of unicorns and leprechauns. It is not a fact. Airborne contagious viruses that attack, multiply, and destroy a living host have never been isolated or proven to exist, let alone cause disease. No one in the world has ever taken just a pure isolated virus particle with no other poisons or toxins or rotting animal flesh added, introduced it into a living organism via a natural mode of entry, like the mouth or nose, and caused the disease that that virus is supposed to cause. Because they have never been isolated or proven to exist, PCR tests cannot and do not detect any such thing as a contagious illness-causing virus in your mucus sample. They detect small partial genetic material from broken down cell fragments found in nearly everything. That is a fact. Genetic material wrapped in protein is neither airborne nor contagious, and the term virus does not mean or describe any such nonsense. You cannot catch a virus any more than you can catch a teenager's acne. That is also a fact. Neither microzyma like bacteria nor viruses, as mainstream science claims them to be, have ever been proven to cause disease. In fact, only disproven in the actual experiments done to try and prove this. What gets confusing is that even in the camp of the opponents of virology, there seems to be confusion over what viruses even are. Though both camps agree they are neither harmful nor contagious, they activate white blood cells to remove dissolved waste debris from the body when the body is too toxic for its own bacteria to do that job. One camp says, viruses are nothing more than dead cell debris. Our cells can die naturally before reproducing. They can die from nutritional starvation. They can die from acidic fermentation because our body pH is too acidic from poor diet or mental worry and stress, which changes our body's chemistry. And they can die from external poisons like heavy metals and gases from chemical and pesticide-laced food sources or bad air pollution, ingesting toxic pharmaceutical products, or injections of poisons in vaccines and poisonous animal bites. As that cell dies, Its DNA and RNA strands break up into thousands of tiny pieces, as do the proteins and other materials that cell was made of. This waste soup of leftover debris ferments, and whatever leftovers cannot be used to make new cells has to be removed from our bodies. Those partial genetic strands of DNA and RNA are wrapped in protein debris from the dead cell, and we call this a virus, just like your garbage bag around your garbage in your kitchen. Virus, dead cell debris. Germs, also known as microzyma, fungi, and bacteria, are dead cell debris eaters. They are our body's janitors or garbage men. They come from inside of our body, not outside, and are there to help eat our dead putrid cell debris, not to harm us. These germs are not attacking these strawberries from the atmosphere. These strawberries are dying, and their internal germs are eating up the dying cells. If they were coming from the atmosphere, the top and outsides of these strawberries exposed to the surrounding atmosphere would be molding first, not the oxygen-starved insides. Eating poop will make you sick, but it is not the germs and bacteria in your own poop that is poisonous to you. It is the toxins in your poop. That bacterial poop goes into our bloodstream and activates our white blood cells. 60% of our white blood cells are called neutrophils, but they do not kill or eat bacteria. They suck them up to reduce this poop in our bloodstream after the bacteria is done eating our dead cellular debris. That's what we call antibodies. Bacterial poop is not supposed to be in our bodies. It is an antibody, and neutrophils simply get rid of all non-self. Your dog eats poop all the time and doesn't get sick because it has a better internal bacterial and neutrophil janitorial system than we do. So it's not the germs that cause toxemia disease. Germs cure toxemia. The other camp claims that viruses are not at first the waste product of cellular breakdown, but are protein-based soap solvents created inside of the cells and released into the cell and the blood to dissolve and clean up waste and toxins in and around the cells when that area of the body is too poisonous for germs to survive.
Viruses are created in the following way. When toxicity has reached a point wherein it can no longer be held back by the body, mass amounts of toxins will be dumped into the blood system. In order for the body to get rid of these toxic accumulations, now in the blood, bacteria come in to feed upon this matter as the first primary method of removal. The processes of phagocytosis play a role as well. Parasites and fungus are also involved in small amounts, depending upon the toxic nature of the substances. However, if the tissues are so toxic and the substances so toxic in the body, living microbes will be poisoned to death when they attempt to consume this matter. The body has viruses to accommodate this situation. In such circumstances, cells come together as a whole unit, conspiring to cleanse themselves and their surroundings of toxicity. In order for a cell to manufacture a virus, it does so in the following ways. First, the cell will call upon the viral protein already existing in the cell. The genome plays a part, and the cell will call upon the blueprint of whatever virus is needed and determined by the cell to cleanse itself and its surroundings. The viral protein is called into the nucleus, wherein the virus is structured into its whole form. Before, all that existed was viral protein particles. Now, the whole structure of the virus has been manufactured and assembled in the nucleus. It leaves the nucleus where it is housed within the cell. The virus will obtain its RNA or DNA by the cell. Since the cell contains DNA, that means the virus being manufactured by the cell will also impart its DNA to the virus. This process allows the virus, with the help of antibodies, which are a type of white blood cell, to dissolve specific tissues. Replication continues in this way until the cell can no longer hold the viruses in itself. At this point, the virus wall will rupture on one side, allowing the viruses to escape and enter the rest of the body. The cell, while ruptured, is not destroyed in the process. The cell can easily heal this small rupture. This set of viruses will continue for 72 hours. After 72 hours, the cell will manufacture a new set of viruses with different cleansing mechanisms. After the first set has been exhausted by the body, the second set will be a little bit different than the first set, but both sets are the same type of strain. The second set will cleanse what the first set did not. When viruses break down large amounts of toxic matter, that matter must be expelled by the body somehow. It must come out of the body. It uses mucus, coughing, sweating, skin excretions, and bowels to remove them from the body. If it is a respiratory virus, there will be respiratory symptoms to expel toxins from the lungs.
Now let's hear the germ theory cult's explanation for bacteria, microzyma, viruses, and disease before we spend the next 2 hours thoroughly debunking it.
During a viral infection, viruses inject their genetic material, DNA or RNA, into a host cell.
That is not a virus. That is a bacteriophage.
In doing so, they reprogram the metabolism of the host cell so that it produces viruses from its own cell material. Viruses can only reproduce in the host cell. They do not have metabolisms. Therefore, they are not living creatures. They are not living creatures. They are not living creatures. Bacteriophages infect bacteria. After attaching to it, the virus injects its genetic material into the bacterium,
A not living creature like a coffee cup, with magical intelligence to know when it's attached to a bacterium, and with a magical physical animation mechanism to inject its genetic material into a bacterium. That's one smart corpse. Must be a zombie.
which starts producing viruses. When released from the dead bacterium,
And what killed the bacterium? The genetic material it was happy to reproduce? It wouldn't happen to be the toxic blood it's floating in, would it?
the viruses start infecting other cells. The protein shell of the influenza virus contains RNA, and this protein shell is surrounded by a lipid membrane, which derives from the cell membrane of the host cell. Influenza viruses spread in tiny droplets caused by coughing and sneezing.
And when and how exactly did this alleged dead thing turn into this dead thing? Decide to start attacking cells instead of bacteria? And how did this dead thing penetrate into a cell membrane designed by nature to keep this thing out?
When reaching the airways, they inject their RNA into cells.
So we have another dead inanimate object that injects its RNA into cells through its own protein coating and through a cell membrane with no injection mechanism. Do continue.
The host cells then produce viruses, which infect further cells. The infection is usually harmless, but in certain cases it can lead to life-threatening complications.
Come on, we weren't born yesterday. What are you selling?
Vaccination is an effective method of preventing influenza. Due to the high mutation rate of the virus, it is recommended that vaccinations be repeated every year.
Every time an animal gets run over by a truck or a car,
we find a big pile of stinky, rotten, poisonous guts putrefying in the road. Later, either the birds come and eat those guts, or a road crew comes along and cleans up that putrefied roadkill. Germ theory and virology claim that the putrid animal guts themselves and or the birds and the road crew killed that animal, not the car that ran it over in the first place. If you are drinking toxic water full of bacteria poop, it can make you ill, just like drinking a bottle of vodka can. But vodka is not contagious. Why are many of the symptoms of poisoning remarkably identical to those of the seasonal flu, the alleged coronavirus, and adverse vaccine reactions? Medical researchers have chronicled over 20,000 different diseases. They name almost every variation. They only have thousands of disease names because of the number of bodily organ and tissue systems that express the symptoms of chronic poisoning and poison expulsion from the body in thousands of combinations of different ways. All of this is only one disease, and that disease, which we call constructive disease, is occasioned by the body itself and is known as toxemia or toxicosis. You cannot vaccinate against toxemia or toxicosis any more than you can vaccinate against getting run over by a car. That is a fact. Pointing fingers and screaming at each other over whether vaccines are poisonous or not is tantamount to fighting over whether unicorns and leprechauns are malevolent or benevolent. Who cares? Vaccinating against toxemia is as pointless as brushing your elbows every day to prevent tooth decay. A healthy body and mind that is not malnourished and or deficient in proper vitamins and mineral cellular tissue salts or over-acidic in pH from poor diet and mental stress from constant fear or worry can easily handle and remove most poisons, toxins, and waste products inside the body. That is a fact. You are hopefully asking, where is the proof of these facts? What is COVID-19 then? Why is the world locked down and society destroyed over a theoretical virus that doesn't even exist? And most importantly, what then are all of these virologists and big pharma corporations actually doing? And why do they believe in their own hocus pocus? 97% of all scientists agree with those funding their research grants. As Dr. Dennis McCohen simply stated, medical students never get to the stage of asking questions. Let them ask one and see what happens. My local university library is divided into two main sections: the medical library, where medics can consult authoritative textbooks on all branches of medicine, and the general library for everybody else. Significantly, all works relating to the sociology of medicine, the critique of medicine, or medical history belong to the general library, where medical students will not have to be exposed to the possibility of reading books that might actually question the premises of the system in which they are being trained. As Arthur Furstenberg pointed out in his book, The Invisible Rainbow, numerous experiments seeking to prove direct contagion through close contact, droplets of mucus, or other processes have proved fruitless. From 1933 to the present day, virologists have been unable to present any experimental study proving that influenza spreads through normal contact between people. All attempts to do so have met with failure. Dr. E.W. Cordingly points out, medical doctors are working on the germ theory of disease, not fact, but the germ theory is already weakening and is due for being thrown aside.
Dr. Fraser of Canada and Dr. Powell of California have experimented with billions of germs of all varieties, but they have been unable to produce a single disease by the introduction of germs into human subjects. Dr. Waite tried for years to prove the germ theory, but he could not do so. During the World War, an experiment was conducted at Gallops Island, Massachusetts, in which millions of influenza germs were injected into over 100 men at the government hospital, and no one got the flu. Germs are scavengers.
Well, I'm glad you brought that up because there is a book called The Invisible Rainbow by Arthur Furstenberg, and it describes that right after the Spanish flu pandemic, which, by the way, resulted in 50 million deaths.
Right.
Okay, so that is way, 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 way more than our current situation. But the U.S. Public Health Service really wanted to get to the bottom of how this disease is spread so that if there was a future outbreak, they would know how to contain it. So they did a series of 3 experiments with people. They did some additional animal experiments as well. But the experiments with the humans, they had 100 healthy subjects, and then they had several people who were sick with the flu. So what they did is they had the people who were sick with the flu cough up as much nasty stuff as they could. They put swabs in their nose to collect the snot. They even swabbed their eyeballs. So they were trying to collect all of the particles that they thought were infectious that they could then deliver to a healthy person and make them sick. So in the first experiment, what they did is they squirted these secretions into the nose, eyes, and throat of the healthy people. And out of those 100 healthy people, not one of them became ill with the flu. So they thought, well, we need to try harder, and they devised a second experiment. In the second experiment, they took the same secretions from the sick person who was coughing and sneezing and tearing, and they then injected that into the healthy subjects. And once again, none of the subjects became ill with the flu. So they devised a third experiment thinking that perhaps a more natural, casual human contact is required for transmission of the disease. And so what they did is they had the person who was sick with the flu sit really close, like uncomfortably close to the healthy subject. And they had a conversation for like 5 or 10 minutes. I forget the exact time. And then after that, they had the sick individual take a deep breath in and the healthy individual take a deep breath out. And they put their lips together almost as if they were kissing, but no contact. And the sick person blew all of his breath into the healthy person who inhaled it all. And the results of that experiment once again showed that none of the healthy subjects became ill. So I've tried very hard to find additional experiments where they tested this theory about contagion of infectious agents, and I could not find any conclusive studies that proved contagion exists.
Pandemic. Why viral? Because the people are poisoned, they excrete toxins, they look like viruses. People think it's a flu epidemic. In the 1918 epidemic, the Boston Health Department decided to investigate the contagiousness of this. So they, believe it or not, took hundreds of people with the flu and they sucked the snot out of their nose and injected it into the healthy people who didn't have the flu. And not one time could they make the next person sick. They did this over and over again, and they were not able to demonstrate contagion. They even did it with horses who apparently got the Spanish flu, and they put bags over their head, and the horses sneezed in the bag, and they put the bag over the next horse, and not one horse got sick. You can read about this in a book called The Invisible Rainbow by Arthur Furstenberg, who chronicles all the steps in the electrification of the earth and how within 6 months there was a new flu pandemic all over the world. And when you hear the normal explanations, how did it go from Kansas to South Africa in 2 weeks. So the entire world got the symptoms at the same time, in spite of the fact that the mode of transportation was horseback and boats, and there's no explanation for it. They just say, we don't know how that happened.
As Dr. Eleanor McBean pointed out in The Poison Needle, during the 1949 polio epidemic, an elaborate study made by the New York State Health Department was not able to establish contraction of polio by contact with other polio victims. The U.S. Public Health Service was also unsuccessful in their same attempt, finding that polio was not contagious. The idea that viruses entered through the nose and mouth led to further experiments on both animals and prisoners, and even orphans in institutions, who were both exposed by close contact with polio patients, and their throats and nasal passages were frequently swabbed with the mucus from the patients that was supposed to contain the polio viruses. No polio was produced this way in either the animals or the prisoners. Polio was not contagious. Not one single experiment in history has ever proven contagious viral or bacterial disease by either injection or close contact. Every single experiment has disproven both, and yet people are wearing 2 masks and a visor, standing 6 feet apart from each other, scared shitless of catching a less than dust-sized nanoparticle with sneeze of a dust-sized theoretical virus. That is a mental disorder that psychiatrists call germophobia. We have all been given a very real mental illness by fearing a make-believe contagious illness. As J. Krishnamurti said, it is no measure of health to be well adjusted to a profoundly sick society. Getting back to the one and only true disease, toxemia, which is not contagious, the great Dr. T.C. Fry tells us on the nature and purpose of disease. In inflammation, the poisons in the body have usually been concentrated in an organ or area of the body for a massive removal effort. That area becomes inflamed due to the constant irritation of the toxic materials there. Our internal janitorial microzyma come to those organs to eat away the toxins and cellular debris from the damage, and doctors are trained to foolishly give us antibiotics to kill off the one thing trying to heal us, blaming the germs for the inflammation instead of the toxins they are trying to remove, blaming the bird for the roadkill and not the car that hit it. When inflammation exists, we are said to have an itis, appendicitis for toxic accumulation in the appendix, tonsillitis for toxic accumulation in the tonsils. Tonsils are the body's first line of defense against orally ingested toxins, most especially heavy metals from braces and tooth fillings, and these cult doctors are foolishly trained to remove them to protect us from viral infections. The names of itises are usually named after the organ or tissue area that is inflamed. Thus, if we have a cold, we have rhinitis. Inflammation of the sinus cavities is sinusitis. Inflammation of bronchial tissue is bronchitis, and so it goes. These are not contagious germ infections. We have these pathologies because in each case, the body has elected to eliminate the extraordinary toxic load through the organ affected. For instance, asthma exists because the body has selected the bronchi as an outlet for toxic materials. The condition is chronic because the toxic condition is unceasing. While the sufferer continues to intoxicate himself, the body continues to eliminate the overload through the bronchi or alveolar tissue. That is why people living in high humidity areas who have chronic lung disease from retaining water in their lungs due to a dietary deficiency in potassium chloride move to dry, non-humid places like Arizona. It's not to escape lung viruses, it's to escape the water in the air. When you see firefighters at the scene of fires, Do you assume that firefighters must be the cause of fires because they are always there? When you see windows in helicopters and airplanes, do you assume that windows must be the reason they are able to fly, since we always find them there? When you see you are a 70% genetic match to a marine acorn worm, do you assume you are no different than a sea worm? Of course not. That would be stupid. Yet virology and PCR tests are based on the exact same stupid logical fallacies, and we don't even raise an eyebrow. Mark Twain said, "It's easier to fool people than to convince them that they have been fooled." The idea of inoculation goes well back into the 1700s, and vaccination started with Edward Jenner in the late 1700s. We've already covered the real reason for the pox epidemic of that time, and Jenner's fallacy in great detail in the last 15 minutes of Biochemistry Debunks Corona. The scope of this film is to pick up with the fathers of modern virology, since the cult is still using their reasoning today, not Jenner's. Coincidentally, this crooked science starts with a scientist named Crook, who in 1885 found strep bacteria in the throat of those who died of scarlet fever. Just like firemen at the scene of the fire or windows in airplanes, with no scientific reason or proof, Crook just assumed that those bacteria were the cause of the disease and death, since they were there. Louis Pasteur found bacteria in the saliva of sick people in 1881, and again, with no scientific reason or proof, just assumed it was the cause of pneumonia. This is still the basis of modern virology. Autopsy a dead sick person or animal, cut out the organ, just as Dr. T.C. Fry said, always an organ of bodily toxic waste and dead tissue expulsion. Place it under a microscope and see bacteria, then finally conclude by baseless assumption that the bacteria was the cause of the problem, not there to eat the dead tissue. Of course, back then they didn't have microscopes that could see anything smaller than bacteria. Nowadays they can see much smaller particles of cell debris that they call viruses. So the blame game is still the same, they've just changed the scapegoat size and name. Correlation does not mean causation. This is no different than saying roosters crow at the rising of the sun. Therefore, rooster crowing causes the sun to rise. Around this same time, 1884 to 1890, sensing the quackery afoot, Robert Koch, or some pronounce Koch, saw the same obvious flaw you see and came up with Koch's postulates, a very simple and logical way to check the claims of people like Pasteur and Crook. More importantly, we need to discuss control groups, or what is today called the placebo group, as well as the gold standard of scientific testing, which again was not and is still not being done to this day, over 135 years of virology fraud. The first step of Koch's postulate is that you must find the microorganism that you suspect or are accusing or alleging is the cause of the disease in abundance in all humans or animals that are suffering sickness symptoms from that disease, whether it's an alleged COVID-19 virus or bacteria in strep or pneumonia tissue. But that same suspected microorganism should not be found in healthy animals or humans. In other words, you cannot be asymptomatic, meaning not sick, and have that organism inside of your mucus or tissue sample. Otherwise, that organism cannot be the cause of that disease. Either that microorganism makes you sick or it doesn't. That is what cause means. Also, if you test somebody who is sick with the symptoms you allege to be the disease, and they do not have that same microorganism inside of them, then you cannot say for certainty that that is what is causing either of them illness. The second step of Koch's postulates is that the microorganism, alleged virus, bacteria, whatever, must be completely isolated from all other tissue and debris in the diseased human or animal sample and grown in a pure culture, not a nutrient-starved and chemically poisoned culture. Then in your control group, you have to purify the sample of a healthy human or animal that does not have that alleged virus or bacteria microorganism in its sample and grow that in a pure sample as well to make sure that microorganisms don't grow there later. This is the only way you can prove germ theory allegations that those microorganisms are contagious invaders from an outside environment and not the body's natural bacteria and microzyma eating the sick dying cells of someone suffering toxemia inflammation and leaving little normal spike protein virus particles and genetic debris as a normal result of the material breakdown of those dying cells. What is most shocking is that to this day, virologists have never isolated what they are claiming or alleging to be a full virus microorganism pathogen, even though first-year medical students all do this very easily with bacteria. They have never grown one of these alleged viruses in a pure, nutrient-rich culture without adding sick, rotten flesh cells, starving them of nutrients to near death, and then adding antibiotics and poisons to the culture to sicken those cells even more. Nor have they ever run a control test or a gold standard from a healthy individual's cells, also starving those healthy people's cells of nutrients, adding antibiotics and poisons to that culture to make sure the exact same so-called virus or bacteria doesn't also grow in that healthy culture as well, to make sure it is their alleged virus from the sick person's sample that is killing those cells in the culture, and not the act of them starving and poisoning those cells that is killing those cells in the culture. Virologists don't see viral spike proteins in the cell culture before they starve and poison the cells. And after the starvation and poison kills the cells, and the cells break down into debris and spike proteins, they say, aha, there it is, the virus, and it's multiplying. No, the dying cells are breaking into more and more particle fragments, and the protein-destroying enzymes like lysine that they poison the culture with are eating holes in the cell walls and making the protein surfaces look like little spikes, or what they call spike proteins, as it breaks apart and deteriorates. When your AstraZeneca COVID-19 vaccine insert says chimpanzee adenovirus vector encoding the SARS-CoV-2 spike glycoprotein produced in genetically modified human embryonic kidney cells, what they mean is that they took monkey kidney tissue cells, starved them and poisoned them in a culture, waited for that flesh to rot and break down into spike glycoproteins, mixed that rotten flesh debris with some living human embryonic kidney cells, which is why they call it a live or attenuated virus, and injected rotting, poisoned monkey kidney cell debris and some fetus's kidneys into your bloodstream. So now your own bacteria have to come and clean that poison and non-self out of your body, and your white neutrophil blood cells now have to come and clean out that bacteria poop with alleged antibodies. And hey, now you're immune from catching unicorns and leprechauns. If you wanted to find out if eating these filthy, water-soaked raw oysters causes food poisoning, would you have your test subjects only eat these oysters? Or would you have them also drink 2 bottles of vodka and ingest cyanide poison and then blame the raw oysters for making them sick? Because that is tantamount to what virologists do in the Petri dish, and somehow we accept this as valid scientific method. But back to Koch's third postulate. You would then have to inject that purified and cultured alleged virus or bacteria with no other additional poisons or toxins into a healthy human or animal, and it must cause the same disease, sickness, and symptoms as found in the human or animal that the original sample was taken from. Not only that, if you do add additional poisons or toxins, you must again have a control group or a gold standard who are also injected with those exact same additional poisonous toxic adjuvants or antigens, but without the alleged virus material or bacteria, and make sure they too do not get the same disease, sickness, and symptoms, to make sure it was the alleged virus or bacteria or filthy raw oysters and not the adjuvants and antigens or 2 bottles of vodka and cyanide that was making them sick. Remarkably, this too has never been done in virology. And finally, Koch's fourth postulate states that the alleged virus or microorganism must be recovered from that second injected human or animal host, re-isolated, which is impossible since they've never isolated the first virus in the first place, and identified as being identical to the original specific alleged or accused causative agent, to make sure the additional poisonous adjuvants and antibiotics they added to the petri dish in their bogus process aren't a new and different causative agent. The same thing goes for the alleged COVID-19 virus. In order to even test this, let alone fulfill Koch's first postulate, we must have reason to suspect that there is some brand new, never before heard of disease with unique symptoms, different from all other already known diseases, caused by a new suspected and alleged causative microorganism never before heard of.
You could just shout out, what are the symptoms of COVID-19? What? Fever, cough, shortness of breath. Good. Fever, cough, and difficulty breathing, right? I mean, I know that a lot of symptoms have been attributed to this. We can't ask mice if they can smell things. But the respiratory symptoms are the main thing, right? This causes a type of pneumonia. Right? It causes respiratory distress syndrome, right? So there's got to be pulmonary symptoms. So shortness of breath, cough, sputum, like coughing up stuff, sputum production, something like this, right? And then fever. We all know fever, right? Those are the most common things. Okay, they're taking temperatures of people everywhere for this, right? So we'd expect at least fever.
Since that describes every single respiratory syndrome known to man since the dawn of mankind, how do you know you have COVID-19 and not the seasonal flu or Middle East respiratory syndrome for that matter? Well, then you'd have to find, completely isolate, and genetically sequence in its entirety and never before seen alleged or accused disease-causing pathogen in a document on the U.S. Food and Drug Administration website from the U.S. Center for Disease Control, or CDC. All the way down on page 42, we find this shocking statement, literally admitting that when designing their PCR test to detect COVID-19, that no virus isolates of COVID-19 were available at the time the test was developed to even know what genetic sequence to test for. So they used computer database stocks of other already known RNA substances that are not COVID-19 and mixed that into a soup broth containing other genetic material from human A549 cells, which are alveolar lung cells with cancer, or rotting, dying cells, and then mix that with a viral transport medium, or VTM,
which is literally a soup broth made of the genetic material of veal, bovine, and cell-killing antibiotics and antifungals, like gentamicin and amphotericin, to mimic genetic material found in the sputum of sick people with suspected COVID-19. If you are designing a test to detect an alleged bat coronavirus, why in the hell is it made out of human lung cancer cells and sick cow cells that are poisoned by antibiotics? This fraud is literally tantamount to you trying to prove that a new exotic cheese exists by testing chocolate milk to see if it has dairy in it, since cheese does too, and then claiming that chocolate milk is actually an exotic new cheese. That's virology. In fact, Freedom of Information Act requests for proof of the COVID-19 isolated virus have been sent by researchers, doctors, and lawyers to over 19 health authorities in Canada alone, as well as multiple health authority departments in the United States, New Zealand, Australia, United Kingdom, England, Scotland, Wales, Ireland, Denmark, Spain, the European Center for Disease Control, the Netherlands, Slovenia, and half a dozen more, asking not only for their own studies, but any studies or information, and not one single agency has responded that any such thing as an isolated COVID-19 virus exists or has ever existed. We will leave links to all of these Freedom of Information requests or responses in the comment section so you can read them all yourselves. One of the reasons we all believed there was some new and terrifying virus going around is because we were shown frightening images of people in Wuhan, China, falling down in the streets for no reason, or shaking and convulsing uncontrollably in hospital beds. And yet, according to Koch's first postulate, that should mean the rest of the world's millions of alleged COVID-19 positive cases should be falling over and convulsing as well, not have a fever, cough, shortness of breath, or even asymptomatic, meaning no symptoms at all. That cannot be the same disease. So how did this gross misunderstanding happen? As we've discussed, the cult doctors are trained to blame the bacteria, fungi, and viral proteins found in the lungs of typical pneumonia patients as the cause of that pneumonia, the firemen causing the fire. But in what is called atypical pneumonia, where those known bacteria and viral proteins are not found in the lung samples of people suffering pneumonia symptoms, even the cult doctors know that there are many known non-pathogenic causes of atypical pneumonia, such as inhalation of toxic fumes, solvents, and substances, smoking, the penetration of food, drinks, or stomach contents entering the lungs in cases of swallowing disorders or unconsciousness, water in the lungs, autoimmune reactions, water retention or what is known as edema, prolonged bed rest, heart and or kidney weakness leading to inadequate ventilation and blood circulation in the lungs, and also radiation is known to trigger inflammation of the lungs in cancer treatment, which is indistinguishable from typical pneumonia. On December 30th, 2019, a young ophthalmologist or eye disease doctor named Li Wenliang informed 7 of his doctor friends in Wuhan over WhatsApp that 7 people were quarantined in his hospital with atypical pneumonia and were suspected of having SARS, out of fear from when SARS was blamed for an atypical pneumonia outbreak in 2003 in southern China. Virologists from the Chinese Center for Disease Control caught wind of this and sent a team out to test for short partial genetic sequences from their computer database from what was alleged to be SARS. Only tested 5 of the people and found genetic strands that had a 90% genetic similarity to genetic strands of harmless and for decades known main coronaviruses in bats, without bothering to check for the other common known causes of non-pathogenic atypical pneumonia. They noticed that all of these people frequented wet markets in China that sold bat soup, as do most Chinese people, and concluded it must be the Batman virus. This time, just as back in the alleged 2003 SARS outbreak, the same German professor, Christian Drosten, who fraudulently claimed to have come up with a reliable diagnostic test for SARS, now claimed he had done the same to detect this SARS-CoV-2 or COVID-19, despite having not even been confirmed to be the suspected cause of the cases in Wuhan, or even isolated for that matter. Ironically, Science magazine wrote an article about Drosten entitled, "How the Pandemic Made This Virologist an Unlikely Cult Figure." Nailed it. As virologist and microbiologist Dr. Stefan Lanka points out in his paper, Misinterpretation of Virus 2, Beginning and End of the Corona Crisis, quote, "To date, virologists have never been able to isolate a SARS virus from a patient, bat, or other animal, or in the laboratory, nor have they been able to identify an intact and complete genetic strand of a SARS virus. Although the very simple, standard techniques for determining the length of genetic sequences have been available for a long time, it has not been possible to prove the existence and presence of a complete genetic strand of a SARS virus in any other way." Virologist Lanka tells us, when cells die, they are broken down into submicroscopic particles, some of which virologists arbitrarily, meaning without reason, label viruses. Virologists are supposed to be doing, but are not doing, what bacteriologists and first-year med students do all the time, which is simply take the sputum or snot sample from the sick person, then you pour that fluid through a special mesh nanofilter, just like coffee through a coffee filter, to get rid of as much of the larger-sized rotten flesh, bacteria, and cellular debris from your sample as possible, so that only the smaller-sized viral particles you are looking for get through. Then you put that filtered material into a spinning machine and do what is called density gradient centrifugation, which separates the sample into bands of denser particles on the bottom and less dense particles on the top. Then you can extract only the band you are looking for with a pipette, and the virus is thus isolated and purified from the rest of the cellular particles and smaller debris. Let's say, for example, we have a never-before-known or seen particle we suspect may be the cause of illness. We'll call this one the milk virus, or milkvid-19, since it has the same density as milk. We simply suck out the milk layer only with a pipette, and we know we at least have an isolated particle of milkvid-19. If it's a new, never-before-seen bacteria, plant or animal cell, or virus microorganism that you want to genetically sequence and map out, like our new milkvid-19 virus here, you would have to either use the Sanger method, adding nucleotides and dideoxynucleotides, or you could use the Maxim-Gilbert chemical cleaving method. Both are expensive, but can map out the entire genome sequence of a new particle or organism, even if tens of thousands to millions of base pairs long. But if you already know and have mapped out the full genome sequence of an organism like this little single-cell fellow, who we'll call AGMO, for already genetically mapped organism, and suddenly AGMO now goes into apoptosis, which is cell death and breakdown from irradiation or toxic drugs, and melts into tens of thousands of little partial genetic strands wrapped in protein, cellular debris, and toxic fluids, or what we call viruses, until there is barely any recognizable part left of poor little Agmo, let alone a full DNA strand. And then we want to take his liquefied jelly remains and sequence them against our original DNA database of a healthy Agmo to see if his remains have any mutations from the irradiation or toxic drugs that killed him. Then we can use a shortcut called next generation sequencing, where we put the 20,000 small partial genetic fragments from his dead jelly debris into a computer database and tell the computer to put them together like a puzzle until they match the full sequence of a healthy Agmo, and the computer will fill in any missing pieces or blanks, also showing us where any mutations from the dead partial fragments don't match up with the entire sequence of a healthy Agmo. This only works because we know he was an Agmo, because we recognized him under the microscope, watched him die, and know we have only his isolated remains to match against known Agmo genetic sequences in our computer. The reason we can't use next-generation sequencing or PCR testing methods for our new unknown milk vid-19 virus is because we have no known full genome of it in our computer database. And since there are only 4 nucleotide bases, A, T, G, and C, that make up the thousands to millions long genomes of all bacteria, fungi, microorganisms, and plant, animal, and human cells, we could literally take the snot sample from any person on Earth, plug those tens of thousands of dead cell fragments into our computer, and tell the computer to rearrange those fragments until they match the known genetic sequence of an Agmo, and the computer will do it, even though there is no Agmo at all in that snot sample. That's what next-generation sequencing does. We could even take the genetic fragments of little Agmo and tell the computer to rearrange those fragments into the genome of a banana, and it will do it. That's what it's programmed to do, and that is exactly how cult fraud virologists like Christian Drosten are designing their bogus virus PCR tests. Trust the science? Please. Never mind the fact that we and Dr. Tom Cowan have already pointed out that no one in history has ever caused any disease in any person or animal by introducing into them just a pure isolated bacteria or so-called virus particle. If this is still confusing, simply imagine this. You want me to prove Barack Obama exists by presenting a legitimate photo of him to you because you've heard of him but have never seen him. So I offer you this photo collage of hundreds of different people in different locations that my computer pieced together based on a template of what I told it to look like. Is this proof he exists? Why can't I just show you a real picture if he really exists? Are these women real, or just a collage of random pictures of other people who, when put together based on an overall color similarity, look like a woman? That is exactly the game virologists are playing with every single so-called virus ever alleged to exist, but never isolated or proven to.
Isolation means, and by the way, the reason this is so important is if you don't isolate a thing, remember a virus is a thing like a table or a chair or a frog or a lamp. We're not talking about a thought or a feeling or an interest. We're talking about a thing. All things essentially can be isolated. I can't think of anything that can't. And if you haven't isolated the thing, you have no idea what the thing itself is made of, and you have no idea what the thing does. Isolation means to separate one thing from everything else. And that would mean you take somebody who's sick and you do various simple and very doable experiments like macerate, filter, centrifuge, suck out the band, show under electron microscope you have the thing. Then you can characterize the thing. You can prove that other labs can do the same thing and same procedures and get the same thing. And then you can even see whether that thing causes certain troubles or not. So once you do that, you would know what you're working with, and that's really the whole story. Nowadays, and I see writers and different critics of me and other people saying, but how can you say that that a viral culture isn't a isolation? So here is how they do a viral culture. They take some tissue, usually, sometimes it's just a previous culture. But let's say the best way is they take the snot or the lung fluid from somebody who's sick, in this case. They may grind it a little bit. They may filter it a little bit. That's not purification or isolation. Then they mix that. In the original measles experiment, they mixed it with milk. And you have to remember that the game we're playing here is find the little particle that has a protein coating and a genetic material on the inside. So if you mix it with any other source of genetic material, you have immediately blown the isolation experiment, and no matter what happens after that, it's no longer valid. And when you read the original experiment, which they're all essentially done the same way, they say, quote, after swabbing the throat, the swab was immersed in 2 milliliters of milk. Milk is a source of rich genetic material. They mixed it with penicillin and streptomycin. Nowadays, they mix it with gentamicin and amphotericin, both of which are kidney poisons. And then in the original measles experiment, they mixed it with bovine amniotic fluid, 90%, beef embryo extract, 5%, horse serum, 5%, antibiotics, phenol red, and soybean trypsin. So now, including the vero cells, which is the monkey kidney tissue, we have 5 different sources of genetic material, and then it all breaks down because it's starved and poisoned. That's called putting it in minimal nutrient medium. When you do that and you mix it with nephrotoxic, meaning kidney toxic antibiotics, naturally the kidney tissue will break down and you'll release genetic material from the milk, from the horse serum, from the bovine amniotic fluid and the beef embryo extract and the kidney tissue. And then in the original experiment, they said, you cannot distinguish the breakdown products from this mess from an uninoculated specimen, which means even if you don't start with anything from measles, you get the same result. That should have been the end of virology. In other words, all these genetic particles are coming from the breakdown products of one of these 5 sources of genetic material. We don't know which one, but we know that's the case. Therefore, any cytopathic, meaning breakdown effect on the kidney tissue, must have come from the technique of the culture, not from the original virus. And I would actually say period. So in this experiment, they disproved the idea of pathogenic viruses. They then go on to say that we can't tell the difference between the breakdown products of the tissue culture versus a pathogenic virus. And 63 years later, in that paper I read, they say clearly there is no way a virologist can distinguish the breakdown products of any of these 5 sources of genetic material from a pathogenic virus, period. So anybody who says that a viral culture, which is actually a misnomer because it should be a tissue culture, not a viral culture, says that this is the gold standard for the purification of or isolation of a virus, I can only say simply either has never read what a viral culture is, or doesn't understand what a viral culture is, or again, tissue culture, or they have some reason for not wanting to admit what any human being can see as the obvious fallacy in that argument. And every single paper that people have said that say the word isolation and purification or the characterization of the genome have all started with that type of tissue culture, which I would contend makes the entire thing invalid after that. Polio, the tissue, was inoculated on viro cells, meaning monkey kidney tissue, in exactly the same way I just described. Naturally then, with the addition of the horse serum and the embryonic fluid and all the rest, and the antibiotics and the starving of the tissue, that monkey tissue broke down into thousands and maybe millions of different kinds of genetic particles. So at some point, they started counting those particles. So that particle is simian virus number 1. This one is simian virus number 2. And then they got all the way up to simian virus number 40. And that one may have been more predominant than all the other ones. And so they said either that's a contaminant or somebody put it into the vaccine. Now, a vaccine is a live viral vaccine is simply taking the results of that tissue culture, as I just described, cleaning it up a little bit, getting some of the little pieces filtered out or so, and adding a few other stabilizers and other toxic ingredients, and then just injecting that in people, like children. So there was no intention of injecting a simian virus number anything in people. It's simply that if the goal was to get this thing called a polio virus, and the polio virus is identical to the simian viruses, there is no possible way to get the simian viruses out and leave the polio virus in. So as a result, they had no choice but to inject the whole mess and hope nobody caught on, that they were injecting just the dead and dying degradation products of monkey kidney tissue. Now, that simian virus doesn't cause cancer any more than any other virus, so-called, causes any other disease. If you doubt that, then show me the evidence that a simian virus isolated causes cancer in anyone. Now, I will admit that if you inject the products of a tissue culture, as I just described, into people, they will, in fact, get sick. And they might, in fact, end up with cancer. In fact, I would say that is what we call a vaccine. And I would even go so far as to say that's the whole point of a vaccine, which is to make people sick. And they do it by the injection of the products of tissue cultures. So in a funny sort of way, you could say that worked. They made people sick, but they don't make a spreadable virus. And it's not the simian virus that makes people sick. It's just the results of this toxic tissue culture. What about so-called antivirals, including natural antivirals? Like there's a lot of herbs which are presented as antivirals. What does that mean if I say that viruses don't cause disease, aren't alive, don't reproduce in the way we say, and the whole thing is basically a bunch of baloney? So how do I reconcile that? It's actually very simple. Anything that's called an antiviral, what it's actually doing is protecting the tissues. So in other words, if you take somebody's snot and you put it on a viro cell and then you poison it and then it breaks down, if you put strophanthus extract in that culture, that same amount of poisoning, you'll get less breakdown of the kidney tissue, and then you'll say, oh my God, that was an antiviral. Because you get less breakdown, therefore less viruses, and then you erroneously think that you've inhibited the reproduction or the growth of these viruses. You've done no such thing. What you've done is protect the tissue. Sterfanthus essentially helps you get more oxygen and helps the metabolic processes of oxygen utilization in the tissues. Therefore, you will get less breakdown of the tissue when you try to poison it, therefore less genetic particles, and therefore you will think that that's an antiviral. The same thing happens with vitamin C. There's no virus-killing effect of vitamin C because there's none needed. But vitamin C is a strong detoxifier and protector of the tissue. And if you do that in a culture, you will see less of these particles, and you will erroneously think that you've had an antiviral effect. So that's what we mean by an antiviral effect. Next question, is it possible to isolate exosomes? The answer is yes. And that's one of the answers to people say, well, the reason you have that no pathogenic virus has ever been isolated from a human being is because you can't isolate things that small. And that's simply scientifically incorrect and has been for 70 years. So there's at least 3 things of approximately that size and that consistency which have been regularly, consistently, and easily isolated. And those are bacteriophages, which was what happens when bacteria are stressed. They essentially coalesce into a kind of spore, and they have the same shape essentially in size as what we call viruses. And many of those have been isolated. The giant virus that Stefan isolated is also similar, and exosomes. So the issue is not that we can't isolate that size or consistency particle, it's that they're just not there. Exosome, remember, are just products of the breakdown of our own tissue. They're little packages essentially of genetic material that our tissues or any animal tissues, probably, I don't know for sure, but probably plant tissues as well, make when they're poisoned or stressed. And as far as I can tell, the reason they say they don't do that is it takes way too long. And anyways, this virus was going to kill us all before we could get to that. So now we can do it in 2.5 days in a computer, except they don't do it anything like that, and they're not actually dealing with this, a real virus. And that's why me and Andy and Stefan are so determined that at the end of the day, saying there's no virus, there's no pathogenic virus, there's no SARS-CoV-2 virus, it's never been isolated, viral culture is a fraud, PCR test on something that you haven't isolated is basically a fraud. That is the only show in town, because that's the only way we're going to stop this. Everybody who thinks that there's going to be mutant strange, variant strange, is just buying into the whole narrative, and we're going to be dealing with for the rest of our life.
So we're going to start off talking about this SARS-CoV-2 virus. It's not really a virus, but they call it a virus that is allegedly behind this pandemic. And then we're going to go backwards in time from there. So what did they actually do to prove that this virus exists? Because that's the main proposition that leads to all of the downstream effects, including all the policies we've been subjected to. And what they did is basically they took some lung fluid, right, pus from the lungs of sick people, and from that messy sample, which contains all kinds of junk, right, it's basically rotting flesh from a sick individual, they took out some small fragments of genetic material. And from that, and it seemed like this was their main priority, is to develop a PCR test. So just so you know that they found these small little fragments of genetic material, not just by looking at everything that's there, but they had to look for something specific in order to use this PCR technique to find a needle in a haystack. And what they did was they just took sequences from another virus, SARS-CoV-1, which they also used the same exact techniques to discover. And they said, oh, look, we found similar sequences of short genetic strands, not an entire gene, but little tiny fragments of just 200 to 300 bases long, not enough to do anything. And it's unknown what the significance of those are in reality.
It is important to remember that the average adult loses between 50 and 70 billion cells each day due to apoptosis, meaning cell death, plus billions more from other dying bacteria and microorganisms that live inside our bodies. That dead cellular jelly debris of rotting flesh and acidic waste, which is a virus, meaning poisonous fluid, has to be expelled from our body. Our lungs are one of the main ejection points for all of that rotten, broken down soup of dead, leftover genetic material. It would be a miracle if you didn't find these partial genetic strands wrapped in protein in your sputum. They are not the cause of disease. They are the byproduct of the one disease, toxemia. This virus jelly did not cause Agmo to die. It is the byproduct of Agmo's death from the environment around him. And remember, they're all made of the exact same 4 nucleotides, A, T, C, and G, which is why isolation is so vital. If you want to prove that one of these fragment combinations of ATCG is somehow magically causing disease, while the other billions of fragments of ATCG combinations are not.
When they show images of the virus that are not computer graphics, they're almost always pictures of this where they zoom in and they show particles either inside of a cell or coming off of a cell. And they say those particles are the virus, but how do you know? Because we do know there's lots of different particles that come off dying cells.
I was contacted by a lawyer in Australia, and so he wanted my help in trying to understand the science of virology, and particularly a paper. And this was a very important paper. The title of the paper is The Isolation and Rapid Sharing of the 2019 novel coronavirus, SARS-CoV-2, from the first patient diagnosed with COVID-19 in Australia. Now, there was 2 sections in this paper which really stood out, and I wanted to go over them. So the first one, which is on page 3 of the paper, under the section called Growth, Visualization, and Global Sharing of the SARS-CoV-2 Virus. Electron micrographs of section Viro H-SLAM cells showed cytoplasmic membrane-bound vesicles containing coronavirus particles. And then they said, see box 5B. Following several attempts to recover virions with the characteristic fringe of the surface spike proteins, it was found that adding trypsin to the cell culture medium immediately improved virion morphology. So the first thing is, when they're talking about isolation and characterization, this is exactly the way that I've described it many, many, many times before. These were all done on Viro, i.e. monkey kidney cell cultures, and they never actually isolated anything. But the important point here is they said they looked at the results of this culture from the Viro cells, and they found little vesicles, and they said they contained coronavirus particles, but the viruses did not look like the normal coronaviruses that we are used to seeing. In other words, they didn't have the spike protein on it. And then, quote, it was found that adding trypsin to the cell culture immediately improved the morphology, meaning the appearance of the virus. In other words, they looked through the cell culture and they didn't see any particles with the spike protein. So they added trypsin, which, as you probably know, is a pancreatic enzyme whose function is to digest proteins. which is what the outer coating of a virus is supposedly made out of. So this outer coating of the virus was digested by the trypsin, which then, quote, improved the appearance of the virus. In other words, it was only then that they could see the spike proteins, which immediately would make anybody think that the only reason you could see the spike proteins is because they dissolved the proteins around the coating, which would naturally leave little spiky things protruding out from the membrane.
I'll tell you what we'll do, though. We'll bloody enhance the value a little bit. Give her a f***ing bloody decent mustache. Let's f***ing Chopper Lisa. Chopper Lisa, how are you?
When you hear this, you think you can only shake your head and think, what the heck? Like, that's not how people do stuff. The fact of the matter is, there was no spike protein looking particle from the culture. Forgetting about even that the whole culture thing is a bunch of nonsense in the first place. They simply could not find a particle with spike protein. So they cut away some of the edges, leaving naturally protein, spikes protruding from the wall. And they said, there it is, the spike protein. If that isn't the wackiest way of thinking I've ever seen, then maybe the next one in this article will be even wackier. So the next section of the article is called the phylogenetic analysis. That means it's sort of the natural history of the virus. And so again, a virus is a genome, in this case, supposedly RNA, packaged up in a protein. The genome is approximately 30,000 base pairs long. A base pair is like a letter. So it's like saying a book is 30,000 letters long. And the whole trick for virologists, if they find the genome, they essentially say they found the virus. So they're looking for this 30,000 letter or nucleotide piece of a genome. And so here's what they said.
Phylogeny means of the same species or taxonomy. So phylogenetic analysis means running the Sanger or Maxim Gilbert methods to determine your genetic material sample is indeed from a so-called coronavirus family and not just broken down human, fungal, yeast, bacteria, or bacteriophage cell fragments, or even from a small bug that flew up your nostril.
Phylogenetic analysis indicated that the genome sequence of our isolate exhibited greater than a 99.99% sequence identity with other publicly available SARS-CoV-2 genomes. On the surface of it, one would think that there's a published 30,000 base pair genome from the SARS-CoV-2 Wuhan virus, right? So that's published. So that's like saying we know this book, this 30,000 letters with the periods and commas and spaces, we know the exact sequence, 30,000 long. And then you would think they looked through this mess of 50 million or so pieces of RNA, and lo and behold, they found a predominantly 30,000 base pair sequence in their sample, and amazingly, it had a 99.99% match to the original sequence in the Wuhan virus. Now, that would make you think, naturally, that you in fact found the same genome, therefore you found the same virus, therefore the sample of this Australian patient was the exact same SARS-CoV-2 virus as found in Wuhan, with only a 0.01% approximate variation. That is what any normal human being would think. And if that was the case, that would be a very convincing case that they in fact did find the same virus. However, on investigating what they did, here's approximately what these researchers did. So number one, before the phylogenetic analysis, they had come to the conclusion that this infection, this virus, was a SARS-CoV-2 virus. That's step one. Then they have these, 20, 30, 40, maybe 50 million pieces of RNA in essentially a brew. And then they have to align these small pieces or fit these small pieces into one 30,000 length chain. Now, obviously, you can't do that out of nowhere. So they have to put a template into the computer and essentially say, align these or arrange these series of nucleotides to match the template that we entered into the computer. And lo and behold, you find out that the template that they entered was none other than the SARS-CoV-2 Wuhan genome. So essentially what they did was tell the computer to look through this 50 million or so sequences, small sequences, these are like letters, and arrange them to make the exact sequence found in the Wuhan virus. They did. They made 0.01% mistakes, and then they said, oh my God, we found the same virus. So here's an analogy to help you. So you have 20 million letters, you put them in a box, and you say to the computer, make this into a story. And this computer says, which story? And you can't say, just make it into any story, because the computer can't do that. So you say, here's the story of Hamlet. Arrange these letters to make Hamlet. So, of course, the computer does. And along the way, it makes a 0.01% mistakes. And lo and behold, then you say, oh my God, this is the same play, the same book as Hamlet. It's amazing we found the same play in this mess. Now, obviously, if you had put Moby Dick into the template, the computer would have spat out Moby Dick. If you put Dostoevsky's Crime and Punishment, the computer would have spat out Crime and Punishment. None of them, not Hamlet, not Moby Dick, not Crime and Punishment, none of them could be found in the sample. That was a creation of the computer. Now, if you ask these researchers, and apparently that's what's going to happen, they're going to be deposed and have to answer for this. My guess is the answer they'll give is, well, we already knew it was a COVID-19 case because we found the coronavirus with the spike proteins. Right, except only when you cut away the outside part. But that's their justification for using the SARS-CoV-2 genome as the template. And if that isn't crazy, then I don't know what is. The variants come, at least partly, there's a number of ways they create variants, or the theory of variants. But if you put in Hamlet and you tell the computer to arrange these letters exactly like Hamlet, every once in a while the computer will make a mistake and you'll get an alignment error, which is then called a mutation. And that's why there's something like 496,000 variants, because every time you put it in the computer, you will end up with a different, slightly different genome, because the computer never seems to be able to quite get it right. There's other ways they come up with the variants, but that's the main one. And that's why we have so many different variations of this imaginary virus, because there's been a lot of computer programs making these in silico, otherwise in the computer viruses, because they don't exist in reality.
Reuters News, the world's largest news provider, owned by Thomson Reuters, partner to BlackRock, a stakeholder in both Pfizer and AstraZeneca, is already pushing the false narrative that there are dangerous mutated contagious strains or mutations of COVID-19 that will kill millions more if we don't take their multiple vaccines. They falsely claim viruses constantly mutate, and new mutated strains have emerged in South Africa, the United Kingdom, the United States, Japan, and even Denmark, and the ignorant scientists worry about any major changes to the virus's so-called spike protein. All of those countries have been given the same computer template for COVID-19, or Hamlet. Snot samples were collected from sick people in each of those countries, and since no two people's genetic snot soup is the same, the computers all came up with slightly different ways to rearrange those people's different genetic snot soup nucleotide fragments to write Hamlet. And the difference in the blanks the computers had to fill in are what are being called mutations of Hamlet, or COVID-19, or what we call fraud.
Germs are biological breakdown products. They're cell fragments. They're born out of the cell. Germs are symptoms of cellular breakdown, period. Okay? They break down just like an analogy would be when you're looking in your refrigerator and you're looking at the deterioration of your food, germs appear out of the cellular breakdown. They're not the cause of cellular breakdown, but they're the symptom of cellular breakdown. So germs cannot cause disease, but are the expression of dis-ease, which is a reflection of the environment. So very simply, we don't get old, we mold. When we're talking about, let's say, anthrax, the bacillus, anthraxis, bacillus, a bacteria, that is born out of the cell. E. coli comes out of the cell. Strep and staphylococcus comes out of the cells. So these are symptoms of cellular breakdown that are a reflection of a compromised internal environment. So it's all about the milieu or the terrain.
Yes.
It's everything, and the germ is nothing. Now, even with the corona effect, which I've studied since the 80s, the corona effect and the spiking, that's an aging process of the membrane as these spikes or these crowns come off as a symptom of just a cell going, from a healthy state to an unhealthy state.
Nobody is wearing a mask and running 6 feet away from grandma in fear of catching her old dying age. That would be ludicrous. Yet nearly everybody has been tricked into doing the exact same thing in fear of catching someone else's old dying cells. Let that sink in.
So when we're looking at bacteria or microparticulates or nanoparticulates, viruses not being a germ, but being the waste of cellular breakdown. So viruses are different because the things that are being tested right now, using the RT-PCR tests, okay, they're testing a fragment of degenerative matter that's coming from either human, plant, or animal.
Which all have the exact same soup of broken down ACGT genetic material from cellular decay and death, which is why kiwis, goats, birds, papayas, and apple juice have tested positive for COVID-19. It doesn't mean they're all sick. It just means they all have fragments of the genetic material the computer is looking for.
This is not science. This is fraud. You can easily test grandma for old age or test a teenager for acne. It doesn't mean they're contagious and cause old age and acne. The Karens will tell you, I know COVID-19 is real. I had it. I tested positive for it during flu season. Yes, Karen, so did a Kiwi. That makes you equally as virtuous as a Kiwi and equally as ignorant of virology and PCR test fraud as Coca-Cola. But as Machiavelli said, a sign of intelligence is awareness of one's own ignorance.
Now you can breathe that in or you can eat it.
Yes.
So if you're eating an egg, that has 38,700,000 pathological microorganisms, and I say pathological only in the sense that you're literally, when you eat an egg, you're putting in the anatomical elements of that egg.
Yeah.
That egg, when it's broken down, the particulates, the matter, the microzyma, which is indestructible, goes into your bloodstream and it activates the white blood cells to say, we have, a storm here. There's pollutants and we need to clean it up. And the ones that have been assigned to that are called neutrophils. It makes up 60% of your white blood count. And they go around, and I watch them. I've watched them for 40 years. They don't attack. They're like vacuum cleaners. They take them into their body, literally sacrificing their lives to maintain internal cleanliness of the internal environment. A germ has genetic matter. Viruses do not. Viruses are poison. Viruses are liquid waste, kind of like when you urinate.
Yeah.
That's liquid waste. Not to be confused by some of the particulates that are in there that have genetic matter. So the flu and the cold is nothing more than the body's expulsion of acidic waste, whether it be metabolic, like lactic acid, whether it be respiratory, like carbon dioxide, whether it be, let's say, environmental, which may be titanium dioxide or hydrogen arsenic or carbon monoxide. Maybe it's dietary, which may be a compound or chemical such as glyphosate. These chemicals that they use in farming they're in pesticides or herbicides or insecticides, but these are poisonous chemicals that end up in our food. Germs are the breakdown products, the symptoms, the genetic fragments of cellular breakdown due to an acidic, toxic environment.
And lately, a lot of people have been sending me, once again, studies published in the peer-reviewed medical literature, some of them from Nature, some of them from New England Journal, some of them from Lancet, a variety of them, saying, here's a paper that shows proper isolation, characterization of the genome, and then a few even having proved Koch's postulates. And so you could ask, so where do they get the genome? They get the genome because they take some of these segments, they find that they attach to certain pieces in this brew, and then they put those segments into a computer, and it makes what they call an in silico genome. In silico is a fancy word for theoretical, which is essentially a synonym for the word imaginary, which is essentially a synonym for the word make-believe. So, and they standardize this in silico genome against the first SARS virus, which was made in exactly the same way with an in silico, i.e. imaginary genome of an imaginary virus. In fact, as I pointed out, when a friend did a BLAST search, which is a search of the essentially genetic database, he found that the one of the primer sequences called the spike protein, it matches 93 human sequences and 91 sequences from other microbes and fungus and other organisms. So there's nothing unique about this. They're simply testing for pieces of genetic material, having no idea the origin of this genetic material. So as far as I know, and I know this, I think, as well as anybody could know it, there is not one study out there, there has never been with this virus or many other viruses that has properly isolated the virus, properly sequenced the virus, and if you can't properly isolate it, you can never prove that the virus is the cause of this disease.
You're probably rightly saying none of this even matters if these virologists can't prove Koch's third postulate, which is extracting this starved, poisoned monkey kidney tissue that has allegedly grown these viruses afterward in a culture, inject that into a healthy human or animal, and make that person or animal sick with the exact same symptoms as the alleged COVID-19 disease in question. This is where the next layer of major virology fraud comes into play. Back to Dr. Kaufman.
Right here, they reported the clinical manifestations, which is like signs and symptoms, what we can observe about these animals after they were given this so-called virus. Now, we had three groups, right? The experimental group, that were the genetically modified mice given the actual virus. You have the second group, we have regular mice given the virus. And the third group, we have genetically modified mice given phosphate buffered solution, which is just like water with electrolytes. Okay? Now, look at how many of them had symptoms in these three groups. You see it's low numbers, and it's less than half in all the groups. So I did a little statistical analysis to show if these groups are actually different from each other or the same. In other words, maybe they just all showed some symptoms, and it doesn't matter what you put up their nose. Okay? And that's what the statistic shows. These groups are not different from each other. So right there, the experiment fails, but they didn't do that analysis in the paper. They just focused on the 37% and said it was significant. So let's look at what these clinical manifestations were. So what symptoms did they actually have? Weight loss and slightly bristled fur. Anyone hear news reports of bristled fur in COVID patients? Weight loss also not a symptom of COVID-19 that's reported. It's a very non-specific symptom. So once again, the experiment fails on this count that this is not the same symptoms as the disease in question. Now we go to the autopsy findings. Now, there's really a lack of data here because normally in a health crisis situation, they're supposed to autopsy everyone who dies suspiciously. They specifically reversed this policy worldwide during this pandemic, and I believe to cover up the true cause of death. So we do have two studies that were published that do describe autopsy findings in a case series of people with this sort of pneumonia illness, right, that they're calling COVID-19. And what they found is the same thing, basically. So you see similar characteristics. Diffuse alveolar damage just means to the air sacs where the gas is exchanged. There's damage. There's hyaline membranes, which you can see under a microscope. There's metaplasia, which is a little bit distinctive, and that's like kind of precancerous cells or cells that develop abnormally. And then blood clots. And that was a very characteristic finding, blood clots in all the autopsy samples. So what do we find in the autopsies of the mice? Well, we found interstitial pneumonia with macrophage infiltration. There was no macrophage, that's a type of white blood cell. They weren't found in the human studies. No hyaline membranes were found. No edema, no metaplasia, and no blood clots. So essentially, the autopsy findings didn't match. Once again, this article fails. So what are my conclusions? None of the postulates were satisfied. And then despite taking a toxic liquid from monkey cells grown with infected lung pus and poisonous chemicals being inserted into the mice noses, less than half showed any symptoms at all. And none of them showed symptoms or autopsy findings consistent with COVID-19. So big fail, fraudulent paper.
Of the definition of people dying of COVID. So the case definition is very simplistic. It means at the time of death, it was a COVID positive diagnosis. So that means that if you were in hospice and had already been given, a few weeks to live, and then you also were found to have COVID, that would be counted as a COVID death. It means that if technically, even if you died of a clear alternate cause, but you had COVID at the same time, it's still listed as a COVID death. So everyone who's listed as a COVID death doesn't mean that that was the cause of the death, but they had COVID at the time of death. I hope that's helpful.
The reason everyone is so frightened out of their mind is because we are told that in the last 16 months, 2,760,000 people have died with, not from, COVID-19. While we know 1.5 million people die every 12 months, or 2.1 million every 16 months with tuberculosis, and we don't shut down society or walk around with masks on, 2.7 million is certainly a large and frightening number. In fact, it's 0.0003% of the world's 7.9 billion population. At this rate, it could kill off the entire world in just 5,000 more years. According to the World Health Organization's own website, seasonal flu toxemia detox complications kill 290,000 to 650,000 people every year, which would be 362,500 to 812,500 people over 16 months. The problem is that we know worldwide hospitals have been instructed by health authorities to give COVID-19 PCR tests to patients, and every one of them who tested positive and then died was to be counted as dying with COVID-19, not of COVID-19. But because it is put on the death certificate as a second or third contributing factor, as seen here, they are all counted in the 2.76 million running death count nonetheless. The first obvious problem with that statistic is that we know COVID-19 has never been isolated or proven to exist, never been proven to cause disease or death, and PCR tests are not testing for COVID-19. As Dr. Cowan said, they are testing for partial genetic material fragments in a snot soup that match 90 known common human genetic sequences, 90 known common other bacterial and microorganism sequences, as well as motor oil, Spanish river water, kiwis, goats, birds, Coca-Cola, papayas, durians, watermelons, apple juice, and even blank tests that have never swabbed a human snot sample have come back positive. So in reality, 100% of COVID-19 PCR tests are what can be called false positives, because they cannot detect any such thing as COVID-19, since it doesn't exist and they aren't testing for it anyway. That brings that frightening 2.76 million deaths down to a much less frightening 0. The second obvious problem is that a PCR test is not a diagnostic test. Its Nobel Prize-winning inventor, Kary Mullis, made that perfectly clear. It can only detect what short nucleotide sequence they tell the computer they are looking for, not how much of that material is in the snot soup sample or if it causes illness or not. Again, you cannot take a small drop of chocolate milk and claim it is a wheel of new exotic cheese just because it has dairy in it. This partial genetic material is so small that they need to amplify the material found in the snot soup sample anywhere from 25 to 40 times before it even shows up in the chemical detection method, which is called a PCR cycle. The problem with that is that if you run a PCR doubling cycle more than 25 times, even genetic material sequences you aren't looking for will amplify and show up as what is called false positive PCR tests. Labs worldwide, like MIT here states, were instructed by the World Health Organization to run these PCR tests at 40 cycles, and if nothing showed up, they called it a negative COVID-19 test. As the World Health Organization, U.S. Center for Disease Control, and the Chinese Center for Disease Control all recently admitted, they had to change their 40-cycle spin protocols because they discovered it was even giving an 80% false positive rate for the little meaningless common genetic sequences they were looking for. Health officials from the states of Massachusetts, New York, and Nevada concluded up to 90% of the people who tested positive did not carry a virus. The Wadsworth Center New York State Laboratory concluded that approximately half of their PCR tests, redone at 35 cycles instead of 40, would no longer be considered positive, and that 70% would no longer be considered positive after redoing them with 30 cycles. An appeals court in Portugal has correctly ruled that the PCR test process is not a reliable test for SARS-CoV-2, and therefore any enforced quarantine based on those PCR test results are unlawful. The Infectious Diseases Society of America found that at 25 cycles of PCR amplification, 70% of their PCR test positives are not positive cases, and since the virus cannot be cultured. And at 35 cycles, 97% of the positives are false. PCR is not testing for a disease. It is testing for a specific RNA pattern, and when you crank it up to 25 cycles, 70% of the positive results are not really positive in any clinical sense, since that RNA they are looking for cannot make you or anyone else sick. The labs were all admittedly spinning them at 40 cycles. Let's take a conservative average of all of these false positive studies. 90% plus 50% plus 70 plus 70 plus 97 plus 80 gives us 457, divided by the 6 studies gives us a 76% false positive average. Every single person who died in hospital with COVID-19, remember, nobody has died of COVID-19, was given one of these bogus, meaningless RNA detecting PCR tests to determine they even had COVID-19. So let's take the 2,760,000 alleged deaths and get rid of the admitted 76% false positive average of those, and that gives us 662,203 deaths over 16 months, the same as the 650,000 who die in only 12 months of the seasonal flu on a bad year, and much less than the 812,500 who would die in 16 months on a bad flu season. The U.S. Center for Disease Control tells us through NBC Nightly News that flu cases nationwide are down an incredible 98%. The Irish health chief says fortunately there's no flu this season in Ireland, and that the flu has virtually disappeared in Australia. Fox News informs us the flu is down from 38 million in 2019 to less than 2,000 after COVID appeared. The UK Metro tells us flu cases are down 90%, giving hope that the NHS could dodge a winter crisis. An article in the Journal tells us the overall clinical consequences of COVID-19 may ultimately be more akin to those of a severe seasonal flu rather than to a disease similar to SARS or MERS. On March 19, 2020, the UK government's own website, said COVID-19 is no longer considered to be a high-consequence infectious disease, or HCID, in the UK, due to more available information about its low mortality rates, and thus the UK Advisory Committee on Dangerous Pathogens, or ACDP, is also of the opinion that COVID-19 should no longer be classified as a high-consequence infectious disease. The UK Telegraph reports on April 2021 that the UK Chief Medical Officer Chris Whitty says no more lockdowns. Britain will treat COVID like the flu. And RT News tells us that scientists are baffled at the disappearance of influenza worldwide. These scientists must not be that bright, because it doesn't take Sherlock Holmes to figure out that the big pharma cult has relabeled influenza as COVID-19 by claiming the exact same symptoms and programming a computer to look for a slightly different partial genetic sequence in the snot samples of people who are sick with the flu detox. In fact, to head off those who say this sounds like a conspiracy theory, here is the former health minister of Belgium, Mark van Ranst, admitting openly on camera that that is indeed exactly what they are doing.
I was asked to tell you about my experiences being the crisis manager, the flu commissioner for Belgium, and highlighting the communication aspects there. And then you have one opportunity to do it right. I mean, day one is so important. In day one, you start your communication with the press, with the people, and you have to do it right. I mean, you have to go for one voice, one message. In Belgium, they chose to appoint a non-politician to do that. I mean, I have no party affiliations, and that makes things a little bit, at that time at least, a little bit easier, because you're not attacked politically, majority, minority. That doesn't come into play, and that was a huge advantage. The second advantage is that you can play in Brussels the complete naive guy and get a lot more done than you would otherwise be able to do. You have to be omnipresent that first day or the first days, so that you attract the media attention. You make an agreement with them that you will tell them all, and if they call, you will pick up the phone. When you do that, then you can profit from these early days to get complete carpet coverage of the field, and they're not going to search for alternative voices there. And if you do that, that makes things a lot easier. And then you have to say, okay, well, we will have H1N1 deaths. Of course, that would be unavoidable. I used there Sir Donaldson's quote, where he said that in the UK, by the peak of the epidemic, 40 people would die per day at the end of the summer. So 62 at that time million people in the UK, 40 deaths a day. I worked it out for Belgium. That would be 7 deaths a day at the peak of the epidemic. I used that in the media. 7 Belgian flu deaths per day at the peak of the epidemic would be realistic. That is true in every year, even interpandemically. That is true in every year, even interpandemically. That is very, very conservative. However, talking about fatalities is important, because when you say that, people say, wow, what do you mean? People die because of influenza? And that was a necessary step to take. And then, of course, a couple of days later, you had the first H1N1 death in the country, and the scene was set and it was already talked about.
So let's shift and talk a little bit about the PCR test, a test that is really meaningless. Now, in this situation, since a virus has never been isolated or demonstrated to exist, and by the way, if you look in Off Guardian, there's an article by Torsten Engelbrecht called Why the PCR Test is Scientifically Meaningless. And in that article, he actually corresponded with authors of several of the major papers claiming to isolate a virus and asked if basically any of those labs had purified the virus or had any samples where there was just virus. And all of them replied that they had not done this. So they admitted it in writing, and that's published. So that's a really important resource. Now, Operation Moonshot is a plan in the United Kingdom that's been put forth by the government there And it's been described in the British Medical Journal. You can see the reference at the bottom. And what this plan is about setting up a testing industrial complex, essentially, that is going to ramp up, and by 2020 is going to allow weekly testing of the entire British population. So let's see what the significance of that might mean for the people of the United Kingdom. Okay, so full rollout is what they call it. They're targeting for the beginning of 2021, so only a few months away. And they want to have 10 million tests every day and the entire population tested each week. Now, the British government has put out some numbers about the prevalence of COVID-19 infections and about the estimated false positive rate. And they get that from other PCR tests used in other viruses, which also don't have solid data because there's no gold standard in those tests either. But they gave that a false positive rate of 2.3%. So we'll just use that. We'll go with it since it's put out by their own government.
It is vital to remember that the Orwellian horror we are about to see is only based on a 2.3% false positive rate statistic, when we all know it's really 100%, since COVID-19 doesn't exist, and 75 to 80% false positive rate just trying to detect their in silico common genetic material sequence, which means the societal consequences are going to be 32 times greater than what Dr. Kaufman is about to describe.
Okay, so if we apply this to the entire population, or to 10 million people that we're going to test, we'll see that a 2.3% false positive rate, the false positives will be 230,000. So they could put out a headline, which would be very shocking and worrying, that there are 250,000 new cases in one day from testing 10 million people. Now, if we apply this to the whole week, the entire population, you'll see that 1.56 million people each week just in the United Kingdom will be labeled as false positive. And this policy has consequences based on the testing results. So you're going to be required to have a digital passport, which is going to say what your weekly testing results are. And if they were positive, you're not going to have access to work. You're not going to be able to go to in-person healthcare appointments. You will not be able to attend venues such as the present one. And you won't be able to fly, and they specifically mention flying because that's international travel they're talking about. So 1.5+ million people in the UK each week will be falsely prevented from engaging in these activities based on a faulty test. This is a real problem. It's coming soon, and something needs to be done.
Those who do not learn history are doomed to repeat it. Nobody in their right mind would let a rattlesnake bite them to make them immune from a rattlesnake bite. Instead, if you are bit, you are injected with a possum blood serum that has an enzyme that binds to snake venom and stops it from deteriorating your tissue and killing you through lipid neutralysis. Poison is not the cure for poison, yet millions of people poison themselves and their children every year with vaccines using this same flawed logic, and never bat an eyelid at just how stupid and pointless that is.
Well, that's here again, the live and dead virus. We're going back to definition. Viruses are poisons. It's kind of like what is worse, the bite of the snake or is the venom of the snake? What kills is the poison. Okay, and you can't inject for health. It just doesn't work.
Right.
And it is so unnatural to think that you can inoculate and put in a toxin. So that's using disease or dis-ease to treat dis-ease. So when we understand flu and colds as an expulsion of acidic waste, the idea that you can put in a poison and then develop some sort of immunity by creating an antibody against that particular poison is total nonsense.
To this day, Even the food and dairy industry heat up our foods, juices, and milks, which damages the vitamins, minerals, and nutrients, and denatures the proteins so that they are difficult to digest, also rendering our food supply useless and pointless, all because of Louis Pasteur's fraudulent claims that the bacteria and microorganisms inside of the food are somehow dangerous to us. Pasteur claimed that disease arose from germs outside of the body, that germs should be guarded against, had only one function, are the sole associative cause of disease, can strike anybody, and can only be guarded against by building up defenses. On his deathbed, just before meeting his maker, he curiously admitted, "Bernard was right. The germ is nothing, the terrain is everything." In 1995, Princeton University released Pasteur's own private lab notes and journal with some shocking revelations.
In fact, Pasteur wrote down in his diary, which became public at the Princeton University in 93, that he was cheating, that he was really intentionally killing sheep and saying, look, in public events, look, they died because they were infected and not vaccinated. And he was using sheep not infected, not treated with nothing, and said, look, they are vaccinated, they are infected, and nothing happens. And all sorts of these things I found in his diary.
Herein lies the crux of the problem. Because it was quickly discovered that blaming germs for illness and selling the masses endless vaccinations and pharmaceutical drug remedies could generate unheard of wealth, and because of the political power and prestige for top virologists, and also the eugenics beliefs of the capitalists who took advantage of this, like the Rockefellers and Carnegies, who discovered populations of undesirables could be brought down and kept in check by poisonous injections. From the days of Pasteur until this present day, the works of Pasteur's contemporaries, like Claude Bernard, Antoine Béchamp, and later the genius Gaston Naessens, Gunther Enderlein, Stefan Lanka, Robert O. Young, and others, were and still are suppressed or denounced, and before the Internet, could only be found, if at all, in the non-medical libraries of universities, whereas Dr. Dennis McEwen pointed out, medical students will not be exposed to the possibility of reading books that might actually question the premises of the system in which they are being trained. What these suppressed doctors and scientists did discover would take another 2-hour film just to cover in detail, and is nothing short of an entirely new understanding of biology and the origin of life itself. But briefly, after the invention of advanced light microscopes and molecular microscopy that could see micro and nanoparticles, and the leaps in the fields of biochemistry and molecular biology technology, they discovered tiny, indestructible, eternal micro and nanoparticles found in all living things, plant, human, animal, insect, and microbial, that are both the building blocks of nucleic acid, DNA, and both cellular and bacteria life itself, as well as both the healers and destroyers of sick cells and damaged tissue from toxic, acidic, non-life supporting environments. These particles have many different names: microzyma, proteids, spores, somatids, phages. The Bible calls them dust, and modern medicine and virology know about these particles. But because they are all trained in allopathic germ theory based on people like Pasteur, Virchow, Crook, and Enders, they don't understand the actual functions and origin of these microorganisms, as it doesn't fit into their taught for-profit germ theory model. And understanding this puts their entire industry out of business, and all of them on the welfare line. Antoine Béchamp was a medical doctor of science, doctor of medicine, professor of medical chemistry and pharmacy at Montpellier, a professor of physics and toxicology at Strasbourg, a microscopist and cytologist. Remarkably, Pasteur, the father of modern medicine and germ theory, was just a chemist, not even a physician. Béchamp found these tiny microzyma in large numbers inside the cells of people with tuberculosis, and found they were tiny chemical factories which have the ability to ferment, meaning they can split complex organic compounds down into relatively simple substances. No different than the microzyma in whey, breaking down complex milk structures into kefir, yogurt, and cheese. He found they were alive and teemed with chemical energy, were essential and indispensable anatomic cellular elements which digested, transformed, and assimilated nutrients required by the cells, meaning they don't kill cells or cause disease normally, they help feed and repair them. He tried to kill them, but they were indestructible, even with radiation. They were insoluble in water, composed of hydrogen, carbon, and other atomic elements. They produced nucleic acid, the building blocks of nucleotides and DNA, meaning they are the building blocks of life, not DNA. These living, energetic, nano-sized somatids make the nucleotides that make the DNA that makes the living blood and cells of human, plant, animal, insect, and bacterial life. A far cry and the complete opposite of killing us all through sneezing and breathing them in. He found these microzyma in all forms of life, and found that under certain conditions they transformed into bacteria, first into round coccoid forms, then those would couple into 2 or more units, or they might sprout into rod forms of bacteria. This is called pleomorphism, or what we would call shape-shifting. Béchamp found that when starved of oxygen, these microzymas, meaning bacteria, yeast, and fungal forms, do not die. They go into a state of rest, meaning they return back into the indestructible spores again until a healthy terrain exists where they can sprout back to life again. He found that the microzymas, or germs, in the organs of young people differed biochemically from those in old people. That should have been the end of Pasteur's germ theory right there. If germs were contagious and coming from an external source, then young and old people would all catch the exact same germs. These germs would not be flying around age discriminating. Is this why we're being told children are less likely to catch COVID-19? Because the PCR tests are looking for microzyma nucleotide sequences found naturally in older people? Food for thought. He claimed they are transmitters of heredity, found within the chromatin material of the human sperm, the microzymian granules needed to genetically reproduce all the different cells essential for the reproduction of the human species. But most importantly, he was sure they were involved in the fermentation process and in the production of disease. And he was later proven correct by Gaston Naissance, who filmed this happening in real time with his somatoscope, and later again by Dr. Lanka, Dr. Young, and others. And here is what virologists can't seem to get through their thick skulls. Microzyma germs are no different than the biblical Lord. The Lord giveth, and the Lord taketh away. So let's break it down for these idiots in plain English so the blind stop leading the blind. Here is our blood, full of microzymas, proteids, spores, somatids, phages, bacteria, yeast, and fungus. Here is the blood in a healthy person with an alkaline blood pH, not experiencing COVID-19, shortness of breath, or coughing from acidosis, with no acidic buildup due to stress, fear, or worry, a poor acidic meat and overcooked food diet, vitamin and mineral deficiency, gas poisoning from not opening your windows for 20 minutes a day in the winter to let fresh air in and bad air out, environmental or food toxins and poisons, lack of oxygen from no exercise, wearing masks, smoking, heavy metals, electromagnetic radiation, or damaged tissue from physical injury. This blood has somatids, spores, and double spores, and maybe some bacterial forms, since nobody is a saint. Here is the bacteria-filled blood of someone who starts to become slightly acidic or poisoned from the aforementioned list, and these spores or phages, like the Lord can giveth, bringing the required nutrients to both cells for their regular living and to the janitorial bacteria to perform their cleaning functions. As Béchamp and Naissance discovered, somatid spores can both become bacteria and nourish those bacteria without turning into them, and bacteria can return back into somatid spores. We call both functions bacteriophages. The indoctrinated morons in the virology cult who still believe in Santa Claus and the Easter Bunny call these contagious viruses. This is the last point the proverbial Lord still giveth. And here is the blood of a person so toxic, acidic, and oxygen-starved in their cells that the bacteria start breaking down themselves and bursting into yeast and fungal forms. Even those people claiming the theory that cell nucleuses create hundreds of thousands of soap-solvent viruses from the DNA to clean cells and the surrounding fluids of toxins need to understand that these conditions have only been observed in poisoned cell cultures in a lab, never in the natural live tissue of a sick individual. So that theory, too, may simply be a misunderstanding of the cell's natural enzyme production to eliminate poisons, just like the opossum's blood does for snake venom. And finally, we have the blood of a person who is so toxic, their terrain can no longer sustain life, even microbial. So the microzyma yeast somatids morph again into the final fungal stages to eat away the rotting, dying flesh, no different than a corpse in the grave or a bowl of dead strawberries on the kitchen counter, before returning to the eternal, indestructible somatids they started off as, once the dead flesh is completely eaten away.
And what motivated me was the discovery of an Anthraxis bacillus being born out of a red blood cell in 1994. And then it morphed back into a red blood cell.
Right.
So it came out and it went back as if, in different forms. So this whole etiology is based upon a word called pleomorphism. Pleo meaning many, morphism meaning forms. And so I realized that the germ theory was not a real theory. That is, by the way, it still is a theory. It was not something that foundationally was represented at the cellular level, that things were morphing.
Because modern virologists are trained in bogus germ theory based on people like Pasteur, Virchow, Crook, and Enders, they look at bacteriophages and assume they must be external invading viruses that kill bacteria, because they've never been taught the works of Béchamp, Naissance, and Lanka that prove these phages are shape-changing spores that can both nourish bacteria or come out of dying bacteria until conditions are right to return to life again. They always blame the fireman for the fire or the bird for the roadkill.
It's a kind of mini spore. It's not a spore itself. When the algae realized that there is no condition to build normal spores, which can swim, look to find a good place for itself and grow out again, so they built much smaller particles. And those particles were first detected isolated and characterized in bacteria. And in bacteria, they are called bacteriophages. Phage from phagocytosis, to eat, to swallow. But those structures are not eating the bacteria. This is a misconception. This is already, a very narrowed view to the things. When bacteria are dying, it must be a virus, right? So, but in the end, these are mini spores which can themselves develop again into larger units. This was Enderlein already claiming that the cellular theory of life must be wrong, because he observed that a cellular structure disintegrate into smaller structures. He just could observe in a light microscope, and even smaller one he could not observe anymore. But also smaller structures do materialize out of invisible structures and forming cells.
Just take the vaccine and go to bed early, my fellow children, or Santa won't leave presents under your tree.